Welcome to the fascinating world of droplet microfluidics, where we create test tubes the size of picoliters. Think of droplet microfluidics as creating factory-made pearls of liquid. Each droplet is a tiny isolated reactor, perfectly uniform and controlled. Let's begin by understanding what droplet microfluidics actually is and why it matters. Droplet microfluidics is a microscale technique that allows us to create billions of isolated droplets, each acting as an independent reaction chamber. Each droplet serves as a test tube ranging from picoliters to nanoliters in volume, thousands of times smaller than conventional test tubes. 1 picoliter equals 10 to the negative 12 liters, or about 1 cubic micron in volume. 2 immiscible fluids flow through microchannels. The continuous phase surrounds the dispersed phase, breaking it into uniform droplets. The flow-focusing geometry is one of the two main methods for generating droplets. In flow focusing, the dispersed phase flows through the center while the continuous phase flows from the sides, squeezing and breaking the dispersed phase into droplets. The continuous phase flows from both sides, focusing the dispersed phase through a narrow orifice where droplets form. This geometry produces highly monodisperse droplets at rates of thousands per second. The T-junction is the second major droplet generation method. In a T-junction, the dispersed phase enters perpendicular to the continuous phase channel, forming droplets at the junction through shearing forces. The dispersed phase enters perpendicular to the main channel where the continuous phase flows. Shear forces from the continuous phase break off droplets at the T-junction, creating uniform particles. Droplet formation depends on the delicate balance between viscous and interfacial forces. Viscous forces are proportional to the viscosity mu times the velocity u. Interfacial forces are proportional to the interfacial tension gamma between the two fluids. Viscous forces try to stretch and deform the droplet, while interfacial tension tries to minimize surface area and keep the droplet spherical. When viscous forces overcome interfacial tension at the critical point, the droplet breaks off from the dispersed phase stream. The capillary number is a dimensionless quantity that characterizes droplet formation. The capillary number Ca equals viscosity times velocity divided by interfacial tension. The capillary number represents the ratio of viscous forces to surface tension forces. When capillary number is much less than 1, surface tension dominates and droplets remain spherical. When capillary number is much greater than 1, viscous forces dominate and droplets deform easily. This graph shows how droplet diameter to channel width ratio decreases as capillary number increases. In typical microfluidic systems, capillary numbers range from 0.001 to 0.1. The Weber number is another important dimensionless parameter for droplet dynamics. The Weber number W equals density times velocity squared times length divided by interfacial tension. The Weber number represents the ratio of inertial forces to surface tension forces. When Weber number is much less than 1, the inertial effects are negligible and the flow is dominated by viscous and capillary effects. In microfluidic systems, Weber numbers are typically very small, meaning inertia plays little role in droplet formation. Droplet size can be precisely controlled by adjusting the flow rate ratio. The ratio of droplet diameter d to channel width w is a function of the flow rate ratio between dispersed and continuous phases. Qd is the dispersed phase flow rate, and Qc is the continuous phase flow rate. Droplet diameter scales with the flow rate ratio raised to a power between 1/3 and 1, depending on the geometry. This graph shows the linear relationship between flow rate ratio and droplet size in typical microfluidic devices. Increasing the dispersed phase flow rate produces larger droplets, while increasing continuous phase flow rate produces smaller droplets. Let's work through a concrete numerical example with a 50-micron wide channel. Consider a microfluidic channel with width W equal to 50 micrometers. The dispersed phase flows at Qd equals 0.5 microliters per hour. The continuous phase flows at Qc equals 5 microliters per hour. The flow rate ratio is 0.5 divided by 5, which equals 0.1. With this flow ratio, droplet diameter is approximately 1.2 times the channel width, giving 60 micrometers. The droplet volume is 4/3 pi r cubed, which equals approximately 113,000 cubic micrometers. Converting to picoliters, this gives approximately 113 picoliters per droplet. Now let's examine a larger channel with different flow conditions. Consider a channel with width W equal to 100 micrometers. Now we use a higher flow rate ratio of 0.5. With this flow ratio, droplet diameter is approximately 1.8 times the channel width, giving 180 micrometers. The volume is 4/3 pi times 90 cubed, approximately 3 million cubic micrometers. This converts to approximately 3.05 nanoliters per droplet. Here we compare three typical droplet volumes: 113 picoliters, 500 picoliters, and 3 nanoliters. A fascinating question. Why do droplets naturally organize themselves into regular crystal-like trains? When droplets form, they spontaneously organize into regular trains with uniform spacing, like pearls on a string. The answer lies in hydrodynamic interactions between neighboring droplets. Each droplet creates flow disturbances in the surrounding continuous phase fluid. These flow disturbances naturally push droplets apart, creating regular spacing. The narrow channel confinement forces droplets to arrange in a single-file line. The spacing between droplet centers is typically 1.5 to 3 times the droplet diameter. Channel confinement plays a crucial role in droplet behavior and organization. When the droplet diameter approaches the channel width, confinement effects become significant. The confinement ratio lambda equals droplet diameter divided by channel width. When lambda exceeds 0.5, droplets become strongly confined by the channel walls. Confined droplets take on elongated plug shapes, filling most of the channel cross-section. Confined droplets move faster than unconfined droplets due to reduced wall friction. Droplet microfluidics enables powerful single-cell analysis applications. Single-cell assays encapsulate individual cells in droplets, isolating each cell in its own microreactor. By encapsulating single cells in picoliter droplets, we can analyze thousands of cells individually. These droplets enable measurement of gene expression, enzyme activity, or cellular secretions at single-cell resolution. Droplet systems can screen millions of individual cells per hour, far faster than conventional methods. This technology revolutionizes cancer research, immunology, and stem cell biology by revealing cell-to-cell variation. Digital PCR is another powerful application of droplet microfluidics. In digital PCR, we partition a DNA sample into thousands of individual droplets. Each droplet contains either 0 or 1 target DNA template molecule. Before PCR, droplets are uniform. After PCR, droplets with template glow brightly while empty droplets remain dark. By counting positive versus negative droplets, we obtain absolute DNA quantification without standard curves. The concentration C equals -1 over volume times natural log of 1 minus positive droplets over total droplets. The rate of droplet generation is a key performance metric. Typical droplet generation frequencies range from 100 Hz to 10 kHz. The frequency F equals total velocity divided by droplet diameter plus spacing. At 5 kHz, the system generates 5,000 droplets per second. In just 1 hour, this produces 18 million individual droplets. This visualization shows the rapid production rate, with each line representing many droplets generated per second. Surfactants are essential for maintaining droplet stability over time. Surfactants are molecules that adsorb at the oil-water interface surrounding each droplet. They prevent droplet coalescence and Ostwald ripening, keeping droplets stable for hours or days. Surfactant molecules arrange at the droplet interface with hydrophilic heads pointing inward and hydrophobic tails pointing outward. Common surfactants include fluorinated oils with block copolymers specifically designed for aqueous droplets. Monodispersity is a measure of droplet size uniformity. The coefficient of variation CV equals standard deviation, divided by mean diameter times 100%. High-quality droplet microfluidic systems achieve coefficient of variation less than 3%. This means all droplets have diameters within 3% of the average value. This narrow Gaussian distribution shows the exceptional uniformity of droplet sizes in a well-tuned system. Let's summarize the key scaling relationships in droplet microfluidics. Droplet diameter scales with channel width times a function of the flow rate ratio. Droplet volume scales as diameter cubed, which scales as channel width cubed. Generation frequency scales with continuous flow rate divided by channel width squared. Capillary number scales with flow rate divided by channel width squared. Let's return to our pearl analogy to understand why this metaphor is so fitting. Like pearls on a string, droplets are uniform in size, regularly spaced, and produced with factory-like precision and predictability. Each droplet is nearly identical in size and can have precisely controlled composition. Millions of droplets can be produced with reproducible quality, batch after batch. The droplets self-organize into ordered structures without external intervention. Beyond flow focusing and T-junctions, other methods exist for droplet generation. Co-flowing geometry uses coaxial flow streams, where dispersed phase flows inside continuous phase. Step emulsification uses sudden channel expansion to spontaneously generate droplets. Membrane emulsification forces disperse phase through membrane pores under controlled pressure. In step emulsification, the sudden widening of the channel causes spontaneous droplet detachment. Once generated, droplets can be manipulated in various ways on chip. Droplets can be split into two daughter droplets using bifurcating channels. Two droplets can be merged by electrode-induced coalescence or passive channel geometry. Droplets can be sorted based on optical signals, size, or other properties using dielectrophoresis or other forces. Droplets can be incubated in delay lines or chambers to allow chemical reactions to proceed. Droplet splitting uses a Y-junction to divide one parent droplet into two smaller daughter droplets. Let's put droplet volumes in perspective with everyday comparisons. 1 picoliter equals the volume of a cube with 10 micron sides, about 1/10 the width of a human hair. 100 picoliters is roughly the volume of a bacterial cell. 1 nanoliter is approximately the volume of a typical eukaryotic cell. 1 picoliter equals 10 to the negative 12 liters.